Okay, welcome everyone again to the Spring Cyber Seminar Series on Evapotranspiration, Frontiers in Measurement, Modeling, and Management from Leaf to the Landscape. I'm Stephen Brantley, hosting. Um, just a quick reminder that next week we will hear from Adrian Harpold of University of Nevada, Reno on water vapor fluxes from snow-covered landscapes. The importance of biotic and abiotic mediated processes. This week, I think we have a real treat. We're going to hear from Pamela Nagler at USGS. Um, Pamela grew up in Georgia. She went to college in Florida and studied climate change in the geography department there. After working for several federal agencies like USGS, EPA, Defense Mapping, she actually got to grow cannabis at a USDA Ag Research Service, and she earned a master's degree from the University of Maryland in geography, specifically remote sensing. She then worked in Japan with the Ministry of Agriculture and Environmental Sciences on an NSF appointment. Then she returned to the U.S. and earned her PhD at the University of Arizona in the Department of Soil, Water, and Environmental Sciences. She's since rejoined the USGS in biology and works on semi-arid plant water use issues in the Sonoran Desert, and I will turn it over to her. I will say that I think we should probably keep questions until the end, but I will help guide us through the questions and comment sessions at that point. If something does pop in your head, you can go ahead and type it in the window, and if there's an opportunity, we can discuss it along the way, but I think it's better for organization to keep questions until the end. All right, Pamela?
All right, thank you. Well, I'm going to talk today about wide area estimates of ET by river red gum and associated vegetation in the Murray-Darling Basin in Australia. So even though I grew up in Georgia, my parents told me I couldn't go to college at the University of Georgia in Athens because it was a party school. So I ended up at the University of Florida and did not do any partying at all. So I thought it was really funny because I went to Australia to work with Tanya Doody, my co-author here, and I had a one-year appointment in Adelaide, and I get to the airport with like 12 bags and my son, and her husband and her are there at the airport to pick me up, and they're wearing Georgia Bulldog t-shirts. And I thought, that is so weird. How would they know that's where my home is? Well, it turns out that they learned all about tailgating with Bob Teskey and had bought all of this Georgia Bulldog gear and wore it pretty frequently. So my main co-author on this research is Tanya Doody, and she is at the Commonwealth for Science, Industry, and Research Organization in the Land and Water Group. And it turned out she's like my doppelganger. She's my same age. She has degrees in geography and remote sensing. And she's working in an agency sitting with a bunch of hydrologists, but really doing a lot more with biology and ecology systems and trying to track it with satellite imagery. Edward Glenn is my mentor and still collaborator here at the University of Arizona. And Chris Jarkow is a GIS contractor with USGS that's working with me. Camaldulensis and Armando are in the engineering department here at the University of Arizona, and they are supplying all of the code and processed MODIS imagery so that we could scale up some sap flow measurements on the ground in Australia to look at it with a satellite. And a lot of times we get the MODIS data straight from the DAC, but this guy here on campus has been able to pre-process that data and provide it to us just for our areas of interest or sites in question. So let me see about moving down a slide. So the study sites are in the Murrumbidgee. Luckily, I'm not sure how many of you guys are Australian, but I think it's pronounced Murrumbidgee River, and it's in Yanga National Park in New South Wales. And I don't know if you can see the hand, but over here is South Australia, and where I was staying is in Adelaide, and then you have Victoria and New South Wales. So the Murrumbidgee is a tributary to the Murray River, which is shown here on this large area map of Australia. So I'm going to talk about two publications that have come from our work as collaborators together in Australia. So if I flip back and forth, it's because the first study was done by Tanya to do the groundwork, the field study doing sap flow. And then I tried to scale it, her results of sap flow and water use to the satellite imagery to say what's happening in wider regions and over time. So the purpose of my work was to characterize the water use by the river red gum, or eucalyptus, on the lower Murray-Darling River Basin in Australia using an algorithm that we developed in the U.S. for riparian ecosystems in the arid semi-west out here, semi-arid dryland southwest region of the United States. So our goal was to validate this remote sensing algorithm by applying it to different river systems in semi-arid lands elsewhere. So we were able to help resource managers quantify these environmental science needs in both nations, in the U.S. and in Australia. So our goal was to test the algorithm by scaling sap flow transpiration acquired for red gum on this river, and then predict ET using the MODIS, or a moderate resolution imaging spectrometer, and Bureau of Meteorology weather data. We downloaded about 9 different weather variables from across the river sites here. We got potential ET and temperature and other variables to go with our algorithm that we had developed in the U.S. So we were going to test these different MET variables in our algorithm. So the aim was to quantify water requirements of river red gum for monitoring tree water use, and then scale the plant water use to areas with remote sensing. So the first part of what I'm going to show you is that Tanya's work was to get a clear understanding of the rate of tree water use and estimate water holding capacity of the soil, and then collectively data would determine red gum water use and plant water availability over the length of time that the soil water would store to support trees before flooding was able to be acquired, and look at ecological impacts of their tree health when flood frequency was exceeded by 5 times more up to for red gum. The second part I'll talk about is our algorithm that we developed to scale those transpiration from points on the river to the landscape level. So riparian red gum forests in the Murray-Darling River Basin were comprised of variable proportions of trees and other plants and grasses and had different seasonal wetland areas. These are all important components of the natural ecosystem, and they're all consumers of groundwater, since there was a big need to quantify their water requirements. And we used sap flow sensors that she had collected on the ground to measure ET of the river gum species, and then we used our algorithm from MODIS EVI on the Terra satellite and estimated ET at her field sites over a 1-year study between 2011 and 2012. So she only had about a year's worth of ground data. We then projected our ET by river gum trees, and we had the interspersed areas of grasses and other understory plants between the trees. So we were able to collect that from the satellite imagery and then project that over the years going back to 2000 through 2013 for this whole region around the Yanga National Park, which was where the Murrumbidgee River came in into the tributary of as a tributary of the Murray River. So as we know, water resources development and drought have drastically changed the river flow regimes in the Murray River area, or in the basin. So flooding in this whole region was getting further and further apart, mainly due to the Millennium Drought, which was from the years 1997 to 2010. It turned out that the MODIS satellite was launched in the year 2000 and has collected data every 16 days since then. So it's data that goes back through the majority of the drought up to present day. So quantification of the flooding frequency to support red gum was needed. And sap flow at these 9 sites, like I said, were collected over 2011 and 2012. And I don't know if you can see on the left side, the ones in orange were the ones that were 5-year since they had had a flood, and the ones in green were 2 years since they had a flood, and there was one site that had they hadn't had any flooding for 10 years. So the study was 2 years across the flooded area in Yanga National Park, and the gradient of trees was, like I said, about 2 years apart for flooding, 5 years, and 10 years. In ecohydrology this year, Judy and her colleagues conducted these extensive field surveys. She published it in ecohydrology. From 2010 to 2012 at 9 red gum sites in Yanga that differed in flood frequency, she collected things like sap flow, evaporative loss, and soil moisture to determine transpiration, evapotranspiration, ET, and plant available soil water before and after flooding. Things like tree density, leaf area index, sapwood area, and the number of dead trees were quantified in 2010 prior to the scheduled environmental releases of water at all these 9 sites. And then transpiration and stomatal conductance of red gums was quantified through sap flow on these trees in and around the 9 sites. And direct evaporation from canopies was estimated and combined with sap flow measurements to estimate total ET from individual canopies. So a large natural flood occurred in mid-2010 due to drought-breaking rains, which ended this millennium drought. And the study was able to compare ET of red gums before and after this major flooding event. That study concluded that red gums are able to maintain LAI while reducing stomatal conductance, giving them the ability to tolerate prolonged dry periods. The authors tentatively concluded that a flood every 5 years was sufficient to recharge the aquifer and maintain the trees in good condition. However, the measurements were conducted over such a short time period, really 1 to 2 years, and only within 50-meter square plots. And I'm going to talk about that study in the next slides here, but I wanted to just say that the goal of our present study was to extend her findings over longer time spans dating from 2000 to 2013 in wider areas. And I'll show you that as I go through these slides. So her field methods was to collect soil plant available water with a neutron moisture meter. They had 11-meter depths, and they did 6 to 8 weekly readings. She also had plant available water measured, and she had red gum water requirements for tree water use, plant available water, and mean rainfall. And then she calculated the water requirements as maintenance time. So she had the plant available water divided by ET minus the rain divided by the number of days in the year. She collected sap flux data using Greenspan sensors. She had tree physiology, which was including LAI, sapwood area, and stomatal conductance and sap velocity. And they calculated canopy interception, soil evaporation, and rainfall over the same period that she collected the data. So she has this divided by flooding every 2 years, 5 years, or 10 years. And I think, as I said, she had 4 sites for every 2 years, 4 sites for 5 years, and 1 site for 10 years. So the transpiration rate was about 400 millimeters a year pre-flood, and it was the same thing post-flood. At 5-year intervals, it went from 105 millimeters to 164, and for the 10-year, it went from 9 to 16 millimeters. So transpiration rate dropped considerably as these trees were deprived of their water. So at the 5-year mark, we saw 74% less transpiration than at the 2-year pre-flood mark, and an increase in transpiration from 26 to 41%. At the 10-year mark, we saw 98% less transpiration than the 2-year, and an increase from 2% to 4% due to high summer rainfall. And the LAI here showed no difference between the 2 and 5 years, but there was significant difference in sap velocity pre-flood and not post-flood, and also significant difference in sapwood thickness pre and post-flood. This is a very small plot of the 10 this is basically 5 sites, and it goes 2-year, 2-year, 5-year, 5-year, and then 10-year. And it's showing 3 things. It's showing vapor pressure deficit and stomatoconductance on the right axis, and on the left it shows transpiration. So what we saw is that there was erratic conductance trends pre-flood indicating tree stress, and that tree stress decreased post-flooding in all the classes, 2-year, 5-year, and 10-year flooding, with increase in peak conductance. And we saw that transpiration increased after flooding, and we saw that trees transpired at night. We've seen that on a whole lot of different studies. Trees invoke stomatal control after about 8 a.m. And here's a plot showing those same sites on the ground, 4 2-year studies sites, 5 study sites, and 1 10-year study site. And so here we can see both rainfall, interception, evaporation, transpiration in ET. And what we're finding here is that at the 2-year, the mean ET was an average of 767 millimeters a year, and by 5 years it had almost halved. And we see that there's a potential threshold of about 550 millimeters a year of ET before these plants could no longer make it. So this is actually showing the soil profile depth and soil water at the 5-year interval. What we found here is that we were trying to develop a formula based on plant available soil water and rainfall to estimate the maintenance time of the soil water reserve. At 2 years, we found that was about 15 months, and at 5 years, we estimated that to be about 6 years, 5.5 years for the maintenance time. What we found is that red gums regulated transpiration rates due to suit their flood frequency zone. They regulated those transpiration rates so that they could persist and live, and they reduced their active sapwood and their transpiration rate until the next flood period. Most of them managed after 2 years without water. The LAI, we found that about 0.5 was the indicator of severe drought stress. So in summary, considerable capacity for red gum water regulation was measured. We had under prolonged moderate drought, trees significantly reduced transpiration, and they reduced their sapwood area. Leaf area also reduced after 5 years was exceeded. Subsequent response of sapwood growth and transpiration when water became available, those variables came back to life once they were given water again. Trees refill water reserves after sunset to reduce daily water stress, and water conservation aided by strong stomatal control after 8 a.m. Heavy reliance on soil water in the top meter of the soil profile, and that follow-up floods in 2012 increased transpiration further. No difference between the 2 and 5-year LAI was found, and that presents a threshold from which to monitor the tree condition. And then the ET of 2 and 5-year trees presented a second monitoring threshold of about an ET rate of about 550 millimeters a year. She drew the sketch to look at management implications, showing that at 5 years transpiration declined by about 76%, and in 10 years from the baseline of a 2-year interval without water. She kind of concluded that it took 8 years to return to a 2-year transpiration rate after the trees had been subjected to these droughts. So that's when I went to Australia and took her data and tried to use some remote sensing imagery to say something about scaling her transpiration results at these 9 sites over the region. And one of the ways I did this is that MODIS provides, through their DAC, subsetted data in 250 meter resolution pixels of NDVI, and that's the Normalized Difference Vegetation Index. It's also the Enhanced Vegetation Index, and the 2 indices, NDVI and EVI, have a different relationship with ET. We found that we are typically using MODIS EVI to calculate transpiration rates in our algorithm. So your MODIS pixel isn't square. It's sinusoidal like this, and this is 250 meters. It looks a bit skewed, and here's your Murrumbidgee River, and this is River Red Gum you can see here. And what we did is put a grid over our site of each pixel. We had 4 pixels per sap flow station, and just literally counted the crosshairs that we hit a tree. And this looks fuzzy imagery here, but we have other imagery that validated those are, in fact, trees. You can see some grasses and understory beneath it, and some water in the image as well. So when we did this for all the sites, and we had n equals 4 per site, we ended up with fractional cover at these SAPFLEX sites determined for a 6-hectare area of each. Straight from the DAC, you can print out the NDVI and EVI per site and just kind of get the phenology curve, or the seasonality of greenness from the year 2000 to present. This is 2000 to 2013. And then I took that and threw it into either SigmaPlot or Excel, someplace to look at running the algorithm with Bureau of Meteorology maximum air temperature data in the algorithm, along with potential ET, and then included the EVI from MODIS in our algorithm. And we're able to plot here, the black line on the top left graph is potential ET, and then we have things like our red line indicates what the equation using MODIS and SAPFLOW would have provided for us. And then we adjusted that for the fractional cover in the green ET. And somewhere under there is a yellow line of just basic sap flux. And so we did this, and this is reversed, but we had a relationship between sap flow and our MODIS-based ET, and we were getting R squares of 0.87, 0.84, something like that for our site. So we had a nice correlation. Plotted up for the 9 sites, we have red gum ET measured by sap flux in open circles, and total ET estimated by MODIS EVI method, and the potential ET is part of that equation. And those are the closed circles, so the black dots at the 9 sites. And this was measured for 1 year between July 11th of 2011 and June of 2012. So that's winter to winter there. Fractional cover of the trees averaged about 70%, 69% using the point intercept method on the imagery. And those trees accounted for 72% of the total ET, comparing sap flow to the MODIS-derived ET. So we think that the trees and grass ET rates were about the same per unit ground cover over the year. And here's some statistics in our weather data. We had the 9 variables: temperature, max, min, rain, net radiation, vapor pressure deficit, humidity, potential ET, and flows. And annual rainfall was about 450 millimeters a year, making it a semi-arid ecosystem, very similar to where I am here in Tucson. And we looked here on the left graph, we had the sap flux data regressed against our MODIS ET data for the red gum trees at the 9 sites. And we ran this through the 0 line, but we can see that the 1-to-1 line provides just some scatter here for the 9 sites. And on the right graph, we're showing ET as an annual curve, or the seasonality of this. And we see that sap flow stayed pretty level throughout the summer months, well, summer in Australia. And so we found that the monthly values of red gum ET measured by sap flux, but estimated by MODIS EVI and fractional cover, did have a very high correlation. I think it was 0.9. Yeah, here we go. So the correlation coefficients are provided here, and the relationship between sap flow and our remote sensing was 0.9. I'm going to go and talk about that a little further. When we were comparing total ET by MODIS with red gum ET by sap flow, and regressing that against maximum daily temperature on the left side and daily net radiation on the right side, what we found is that the total ET included the grasses between the trees using the MODIS algorithm, had a steeper slope in response to temperature and radiation than the ET of red gum by sap flow. So you can see here that the open circles are a MODIS predicted ET, and they're steeper in both these regressions with daily temperature, air temperature, and net radiation. So these are flows. There's a mistake in here. We didn't do anything in 2020 yet, but these are 5-year flows, 10-year, and the last year that was measured was 2012. And what you see here is that MODIS EVI for these flows, 2, 5, and 10 years apart, followed the same consistent pattern over a 13-year period. However, they had different starting greenness points. You can see that the ones that were flooded, the circles, that were flooded every 5 years, and then over here on this top line, this had a much higher greenness before these other sites were measured. And that shows the impact of supplemental flooding over this time period. So although flows are needed to recharge the groundwater, in the short term they flood the soil surface and have a negative effect on vegetation, as occurred in this study. But the 9 sites differed in flood frequency, and the sites received supplemental managed flows during this 2000 to 2010 drought. The vegetation vigor, as estimated by EVI, was relatively constant for all sites except for one, or including one that was receiving high rainfall in 2010. So for our 9 field sites, red gum accounted for about 69% of ground cover, and the tree ET accounted for 72% when you compared sap flow to the MODIS-derived ET. What that meant was that red gum and total ET were strongly correlated, but their environmental controls differed. So total ET had a steeper response to temperature and radiation than red gum alone, and that probably, we're presuming, it reflects the difference between the grass understory interspersed between the red gum trees. River flows were negatively correlated with the red gum and the total ET at the field sites, which makes sense. So then we took this a step further, scaled up to like a river reach level. And what we did, what you're seeing here is there's a town here called Balranald, and Yanga National Park starts in this region and is inclusive of this area that we've highlighted. We estimated just on the imagery that tree cover was 72% of this wide area region. So we then calculated mean ET by MODIS imagery, our method that we've used, and we got about almost 800 millimeters a year, from which 65% was red gum, and the remainder was understory grass. So tree cover was estimated by this point intercept method from this Balranald wide area region, and fraction of trees was close to what we found at the individual sites, 68, 69%. The difference between total ET and precip, which is 444 millimeters a year, was then projected over the area of red gum forest, and we found that the difference, 169, was presumably derived from groundwater. This is of the river flow, million cubic meters. So on this river reach, we found that about 17% of the inflow was consumed by red gum and associated vegetation. This is a graph showing rainfall and flows, and we see that they varied widely during the study period. However, the wide area ET projected from MODIS was relatively constant on an annual basis. We still see a mean of about 800 millimeters a year and a standard deviation of about 75 millimeters a year. So this is just greenness, EVI from MODIS over 2000 to 2013, and then run through our equation with the Bureau of Meteorology met data, primarily potential ET and rainfall, and max air temperature, we calculated ET on an annual basis. So that's what you see as this oscillating curve. What we did find was that greenness was relatively constant over the months, and ET varied because it was based on potential ET, which goes down in winter. So we conclude that despite variability in rainfall and river flows, vegetation in ET and red gum forests are relatively stable between 2000 and 2013. This suggests that groundwater supplies were adequate to support the red gum trees, and rainfall and floods were sufficient to support the grasses and other vegetation between the trees. Total ET responded steeply to temperature and radiation, and sap flow from the red gum trees suggested that different environmental controls exist for grasses and trees. Over the study area of 38,000 hectares of red gum forest, we estimated that flows into the Mirrumbungie River had an annual mean of about 1,000 million cubic meters per year. And then what we estimated from the MODIS imagery was that the difference between transpired ET 800 millimeters a year and the precip at 350, the difference is 444, and that's projected over this region. And so we ended up with about 165 million cubic meters of groundwater from the aquifer, saying that that's about 17% of that total inflow of 1,000 million cubic meters. So we conclude that about 17% of the inflow, which is a pretty significant portion of our river flow, is consumed by the red gum forest. And our study, the second study that does the remote sensing, supported Tanya's earlier study using sap flow at the ground sites, in which she suggested that red gum trees were resilient to drought conditions and that a flood at 5-year intervals appeared to be sufficient to maintain healthy stands of trees and associated grasses. So I want to just say that this work was possible because we physically got to sit in desks next to each other and go to the field together in Australia, and it was supported by an Office of the Chief Executive Award granted to us by CSIRO and the USGS. We had a couple different papers on this, and this paper here is the one in ecohydrology that I tried to detail a little bit in the beginning of the study as a setup to what I did with the remote sensing. So thank you. This is Tanya here, and there are other authors, as I said, but we were the ones doing the majority of the work. So thank you very much for listening.
All right. Thank you very much, Pamela. We have plenty of time for questions. If you want to just, let's see. We have a quick question from Susanna. Could you please show the literature again? I think we can just go back to that. Let's see. Maybe you know where the slide is a little bit better than I do, Pamela.
Let's see. On Tanya's work? Or you mean, hold on, right here, I just said, right here, there's two papers.
Right, yeah, right here. I think this is what Susanna's asking for. Yes, that's it. So for the question and answer session, what I'll do is I'll read the question, and just in case anyone is not able to read it or hear what's going on, I will repeat the question, and then I'll also give the presenter a little bit of time to digest it. So we have a question from Steve. What is the typical taproot depth for the red gum?
I don't know. I'm looking quickly through her paper to see. She must have reported that. I have a drawing of it, and I think I can pull it up. I have a picture here. She's got it at 10 meters, between 6 and 10 meters. Am I supposed to write the answer?
Oh, no, that's fine.
Okay.
I think everybody can hear.
Okay.
Everybody should be able to hear you. So 6 and 10 meters
About
is a
6
typical
to 10.
range.
She's got a drawing of it, and it shows the majority of the root zone being between 0 and 6 meters, but it looks like the taproot can go down to 10 meters.
So I have a question. So I work on a species that also has a very long taproot and can access aquifer water, and we've shown with stable oxygen isotopes that it is actually using differential amounts of aquifer water versus soil water depending on conditions. Have you guys looked into doing any isotope work?
That would be great. I mean, we're trying to use isotopes, in the Colorado River Delta in Mexico for our current work that is measuring the effects of the flows, the recent environmental flows to the Delta in Mexico. And we have colleagues of mine have just published something. It's been accepted in wetlands, doing exactly that for riparian species. So nothing on the Murray-Darling, nothing in our work with Tanya.
Let's see, we have one more person typing now. Okay, we have a question from Susanna. Is there any info on how ET derived from MODIS EVI compares with ET from eddy covariance at this or similar site? I think that's a great question, something that popped in my head as well.
Yeah,
So
well, that's I published a lot of work making the relationship between ground-based data with sap flows, eddy covariance and Bowen ratio results, and MODIS EVR methods in the southwestern U.S. So I've done this work on 2 or 3 riparian river systems out here, 4 maybe, San Pedro, Rio Grande, Dolores River, Lower Colorado River. So my studies have all done this. However, Australia has an amazing network of meteorology stations through the Bureau of Meteorology, but finding eddy covariance or even Bowen ratio towers across Australia are very, very difficult. Most of them are located not in these semi-arid regions that I was working in along the Murray-Darling, and there are some on the east and some up in like the rainforest regions and some in the central area near Ayers Rock. But we found that we really, the AusNet operates, a series of towers, but we couldn't find any within the Murray-Darling Basin that were useful for our scaling. That was one of the very first things we did, was try to sort of data mining of all of the flux towers that were available to us to help scale this data, but they did not exist at all for these regions.
So we have a couple of people typing now, but I would go ahead and ask, so if you already had access to eddy covariance data and you wanted to apply the MODIS EVI estimates, how would that be a beneficial thing to do for your system, or?
Absolutely. Well, I should say that because my career is based on it.
Right.
That's what I do. So
we should talk offline later on.
Yeah. The main things that are important there is that your footprint is so hard to match up because you do get some pixel migration, even though it's supposed to view the same place over time. When you're trying to match it up with a very small footprint from an eddy covariance tower, it's important to know that your vegetation is sort of monotonically the same. You want to, the same height. You don't want to have any turbulence layers over your vegetation that are going to change that the eddy system in that footprint, because that messes up some of the scaling. So we did it with riparian vegetation of different canopy heights in structure, but we also had to change our sample size quite a bit. We had multiple towers and the same exact vegetation footprint and percent cover, fractional cover, things like soil salinity, soil texture, distance to open water, distance to groundwater. These things are very important in trying to use scalars such as eddy covariance data. But we did sap flow and tower data at multiple sites that were the same, so we had repeat information in order to scale it.
Yeah, I think that would be very useful. Steve has a question. Do you have evidence of taproot growth to seek water? The Yarra Forest in Washington, I'm not sure if that's Washington, WA, do this.
Western Australia, maybe.
Western Australia, okay. I didn't think there was Yarra in Washington. But so do we have evidence of taproot growth to seek water?
I don't know.
Okay.
Tanya
Yeah.
would be able to answer that, though, probably. You know, I want to make sure her email is, did I provide her email on here? I can just tell you, it's in case anyone wants to know.
Sure, yeah, please.
Okay, it's just tanya.duty@csiro.au. It might be on the first slide, but I can't remember.
And Emily has typed it into the box as well.
Okay.
And we have one other person typing now. Okay, what is the range of groundwater table depth? Do we have a good idea of what the range would be for, say, annual or over 10 years, what the depth could vary by?
Yeah, I think that I have to go back, and I'm looking through her paper. The ecohydrology paper provides that information, I think, but I'm trying to scan it quickly. I'm sure she has depths of groundwater in here for these sites, but I don't see it offhand. But I don't have it immediately where I can find it. Tanya would have the answer to that.
And he says that's okay, thank you.
Mm.
All right, we have another question coming up shortly. Comment, great job, thank you very much, from Steve Burgess.
Oh, thanks. I'm looking quickly to see. I'm sure she has a depth to groundwater. She's got basal area, sapwood area, LAI, and tree depth information, and I don't see her depth to groundwater, but it's a very important part of this. So you'd think that would be something here. She's got stand characteristics, but I'd have to read through the description again in her paper.
Yeah, it's fine. My guess it would be very similar to the taproot depths.
Yeah, 10 meters.
Yeah, or less.
Yeah.
It looks
well, and
like
the picture, I don't know, in one of these things she had a picture of the flooding, not just the one where she was walking through it, but in the beginning of, what these forests look like when they're flooded. So it definitely is between the surface and 10 meters.
So while folks are still on the line, there don't seem to be any more questions coming in. I just wanted to remind everyone of next week's Cyberseminar series, where we'll learn a little bit about snow-covered systems and balances between biotic and abiotic processes. So please join us for that if you're interested at all. And we will see a little bit of data for that talk on from eddy covariance. But I think we're done with questions.
Good. Well, thank you guys for inviting me.
Oh, sure. Yeah, great talk. And I might contact you on the side later on, because I think this is very interesting, and we might actually have a very good model system here for looking at some of the remote sensing applications of ET.
Oh, that's great. I'm expecting to get this submitted to hydrological processes like tomorrow. So
okay.
I'm hoping it'll go through. I worked on it a long time, but it took like a year to settle back in after I came back from Australia.
Yeah, I'm sure.
Yeah, it was pretty laid back down there. So I finally got the results and hope to have it in hydro processes pretty soon.
Yeah, I mean, it seems like it's a good long-term data set in terms of having, multiple types of climatic conditions, a very long drought, different, flooding conditions, different landscape positions. It seems quite interesting.
Yeah. Well, good. Thank you.
All right. Well, thank you so much, Pamela, for being here today. That was a great talk, and we really.
I just want to comment that somebody else just asked a question about the data sets being free, and they are. On the MODIS DAC, it was one of my slides. I listed the link for the DAC.
Okay,
and then the Bureau of Meteorology data sets were free, but that was probably because I was working through CSIRO in Australia. But, if you're not working in Australia and you just want your meteorology data, it's pretty easy to find for free. But the MODIS data is free.
And Emily, are these presentations, are the recorded presentation, are they posted immediately, or is there a lag time?
Yeah, so the recorded presentation will be up on Monday, and it can be found right from the CUAHSI Cyberseminar page on the website.
Okay, so if folks out there missed anything, they can certainly go back on Monday and look at the recorded presentation again.
Absolutely.
Great.
Excellent. Well, thank you, everyone, and we hope to see you at 3 o'clock next Friday.
All right, thanks.
Yeah, thank you, everyone.
Bye-bye.
Bye.